Hello, my name is Abby, and today I'll be walking you through Lab Module 2 of the Yeast ORFan Gene Project labs, which you can find at yeastorfanproject.com, which is linked in the description below. Alright, so for Lab Module 2, I'm at the Yeast ORFan Genome Project, and I'm going to open up my document version of the worksheet and open up the guide as well. And we are going to start by first going to the SGD again and typing in our gene name. And I'll be using MEC1 again as well for this. And I'm going to go to protein and scroll all the way down to the blast.ncbi. And everything should be all set already, and we'll click blast and wait for our results to load. So our results are loaded, and we're going to want to click on the graphic summary tab, and it's going to bring up this section here, and we're going to click on our TEL1 superfamily, and it's going to open up to more specific information about the hits that we get. And we're going to use our first one, which is TEL1 for the name. So we are going to copy in the name and the accession, which is basically just the identification for the gene that popped up. And you want to take the first two hits that appear under this section. And now we are going to copy and paste the description. And that will also be information that's different for each person. And we'll copy the interval, and that is the base pairs that match up for the alignment. So whatever this is being compared to, that's where it lines up in terms of base pairs for your gene. The E value, and now we're gonna go back through and do our second hit. So copy that one. Copy the accession as well. And we have to click on the linked section to copy the description for this one just because it's longer, and yours might be longer or shorter. It just depends on your gene and the results. Copying the interval. And copying the E value as well. Alright, and now we are going to start working with the Tiger database. So I'm going to go and copy and paste the website for that. And there's more information about each specific program in the guide. So for this, we need to go back to the SGD and get our protein sequence, the same way we did for the first section. So we can copy and paste that into the database, and we can start the search. And this one might take a while to load up as well. And here's what it looks like when the results have all loaded. And we want to look for a hit with the prefix of tiger. We don't want any of the PFAM ones that might show up. So we're going to copy the tiger fam model, its description, and you'll have to look into the different descriptions for each result, as they can mean different things and won't have commonality. And then the score as well, and E value. you can also get a GO number and an EC number. I could not find a section for this. I looked through the page and I am just assuming that my gene does not have a GO or EC number. So I'm going to put non-applicable for both of those sections. And now we are all ready to start with our PFAM database. So going to the guide and getting that website again. And we will need our sequence as well, so it's just better, and we'll need it again later, so to just keep up the sequence throughout working on the worksheet would be a good idea. So we had to click under sequence, put ours in, and click go. And now we will be looking for our PFAM family name, which is under that header, and you can copy that, and you want to take the first hit that shows up. And the description. And DE value as well. And now we'll be looking at the predicted active sites, which is non-applicable, and then the HMM from and HMM2. So if yours appears in burgundy like mine does, the whole PFAM hit is not covered by the alignment, so that means that it's possible that the two proteins don't fold in the same way, even if they have a similar alignment and are conserved between the two. So now we'll be looking for the pairwise alignment. So we want to click under show hide alignment, show, and we're going to take a screenshot of that. And placing it into our worksheet to reference. So now I'm going to walk you through what these results mean generally. So if you scroll over any of the sections, it'll pop up and show you what each thing means. So our HMM is currently just showing our alignment, and the match that appears below it is very similar to what we saw in our alignment in the first module. So this blue, the cyan highlighted letters means that they're the same, and the blue means that they're conserved, so they have similar properties between the compared alignments. And then the ones that aren't colored are not conserved, and then in the match, we have the ones that are conserved but not the same have the plus, and if they're not conserved at all, there's nothing below them, but if they are identical, they'll have the same amino acid below. And then the third section is posterior probability, and it shows the confidence that we have with this, and the asterisks would mean that we're 100% confident, the 9 would mean we're 90% confident, and this dot is just representing that there is either an insertion or deletion that would not allow us to make a prediction with that. And that also corresponds to the query sequence, which you can see in the box that shows up. That bright electric green would correspond to the 100% confidence level that we have, which is shown also in the posterior probability. But then once you start getting closer to the red, you're getting closer to 0%. So like this mucky green color, that's 50%. So, and then in terms of that, you can comment on, let residues in your query sequence, the e-value, how conserved or not conserved they are based off of what we got from the PFAM information before and based off of that graphic. So now we are going to work on the domain summary, and we are going to go and click on the linked family that we chose, and we're going to be looking for the family descriptor and number, which is shown at the top. And now the clan name, which you can go into the tabs and click clan, and that'll be in like the first sentence that pops up. So my clan is the P kinase, and then the clan number, which is CL0016. And now we're going to go to the domain summary page, and we'll take some information from there, and that again will be totally different based off of what gene you have, and that's something that you need to take upon yourself to look into. And again, I couldn't find a GO or EC number. That might show up for some people, but for whatever reason, I can't seem to find one for mine. So now we're going to move on to the HMM logo, which is labeled as that tab, and it shows this graphic that you're going to want to take screenshots of. And then you can place it into the worksheet, and you'll want to make sure that you take a screenshot of everything and try and keep it in order as best you can. So in terms of what this means, the height of the stack corresponds to the conservation at that position, and height of each letter in the stack depends on the frequency of that letter in that position. So you would want to analyze how tall things are in terms of, and comparing that to conservation and what letters are or amino acids are prevalent. So for example, we have that one, it's fairly tall, so it's fairly conserved, and it has that really big F, so the vast majority of the amino acids that are appearing at that point is phenylalanine. And you have this really big R, so that's really, really conserved, and the majority of that sequence section appears as that amino acid. And then another one like that, and for example, you have this one with the F and the H, and that one's fairly tall, but it doesn't have one large outstanding amino acid, so that would just mean that the majority of those amino acids for that are highly conserved and have similar properties. And then in terms of that, you can make notes of really significant one, like that really large phenylalanine group. And now we are going to work on the curated alignment. So we want to click on alignments, and then under C, click the HTML, and it's going to pull up this graphic. And at the bottom it has a key about the color coding, what would denote different structures. So we don't have to screenshot that. You can if you want, if you would like the graphic, but I'm not going to do that now, and I'm just going to comment on the structures that are prevalent. So we have all of these S's appearing for mine. So I'm going to comment, and it doesn't look like the S appeared. I think it just got cut off in the video, but the S represents the N or a 5 residue then centered at residue. a 5 residue bend centered at residue I. And then we can also comment on the types of amino acids. So we have this purple color, which means there's a lot of small or hydrophobic amino acids appearing in our alignment. So I'm just going to comment on that as well. And this will be different. You'll have different structures, different prevalent amino acids based on whatever gene sequence that you're working with. So now we're going to work with the protein data bank. And this is a different website we have to go to, so I'm going to copy the link for that. And we'll need our protein sequence again, but you want to make sure you don't get the asterisks at the end of the alignment or the labeling at the top of the amino acid sequence, because this program won't use it for whatever reason. So under sequence, we're going to paste our alignment in, and we want our E value set to 0.01. And we want polymer entities for our display results. And this is what the page will look like once it loads. And we're going to take the first hit, and we're going to take the protein data bank code, which is 5x60. And the name, which is right below the PDB code, right there. And mine is the intact ATR/MEC1 dash ATRIP/DDC2 complex. And for now, we're going to skip the alignment section and just go and get the E value from the page that's currently open. So my E value is 0, and my identities are 1. So while still on this page, you're going to want to click the hyperlinked PDB code to get more information about your protein. And you want to do this before you switch the tab away from what it is currently, and I'll go over that in a minute. And now we're going to look at the literature about whatever protein you have, and under literature you're going to want to click on the orange PubMed box, and that's going to open you up to the paper that figured out the structure of your protein using either X-ray crystallography or NMR, and that can provide more information about the structure of that protein and how it was, how the structure was figured out, and some of that information can be placed into what your unknown protein does. So it would be a good idea to read a little bit about it, and then we have this other little question at the end asking for more information about the protein. And I just took a couple lines from the abstract that I quickly skimmed through to put in that section. And like all of the other things, the information for your abstract versus my abstract is going to be different. So that's another thing that you just have to take upon yourself to look into more. So I'm just going to copy and paste a little blurb about the function of my protein. So now I'm going to change my section from tabular report up at the top and select sequence. But once you select that, you can't go back to tabular report, so make sure you have everything you need from this first page. And there is no alignment anymore. I don't know, the website must have changed. So you can copy the sequence and do an alignment if you wanted to. But you can get the alignment length, well, the protein length of the one that you would do. And mine is 2,368 base pairs. So I'm just going to copy that down And I'm also going to put in the accession number just in case I need it. It's there. You don't have to do this either. But you can always go back and get the sequence and do an alignment yourself if that's something that you want to do and are interested in. Sorry about that last section. I feel like it's a little confusing, but when I originally did the video, instead of saying at the tabular review, I accidentally went to sequence first without getting the literature information, so you can just get that information, click the back arrow, and then switch into sequence and take it from there. But all in all, I hope that you liked this video and that it was helpful. And for more information, you can go to the yeastorfanproject.com. Thank you for watching.